lecture we discussed about the basic concepts of spectroscopy techniques. If you could recall, spectroscopy is mainly the interaction between the matter and the electromagnetic radiation, and electromagnetic radiation in terms of say frequency or wavelength carries particular energy. It is a form of energy which is derived from matter only, and electromagnetic radiation molecules absorb or emit light. They could absorb the light or electromagnetic radiation, or they could emit the electromagnetic radiation, as well as they can scatter also different types of electromagnetic radiation and particles like say electrons or neutrons. So, these atoms or we can say electrons, they reside in discrete energy labels. That is, it will have a ground state, it will have the first excited state in electronic label, and to excite from one state to another state, they require very specific amount of energy. That is the difference of the energy between the two energy states, and once they absorb that particular amount of energy, they could be excited to higher energy level, and that particular information where either it could be absorption or emission could be recorded. Likewise, there are spectroscopic techniques which have developed, which are based on scattering of the particles or electromagnetic radiations by the matter. The wavelength at which maximum absorption takes place is called lambda max, and this particular property, that is the wavelength of light, is specific for each transition. That is transition, as we were talking about movement of the atoms or the electrons, as we can say, to higher labels. This is called transition actually, and this transition is specific in terms of energy, which is related to the wavelength of that particular radiation, type of radiation. So, we also introduced the techniques which we are going to discuss in this section, which included UV visible spectroscopy, IR spectroscopy, circular dichroism, fluorescence spectroscopy, atomic absorption and flame emission spectroscopy, and X-ray diffraction or X-ray spectroscopy. Mostly we will be discussing about X-ray scattering, then nuclear magnetic resonance spectroscopy and mass spectrometry. In this lecture, we will be discussing about one of the techniques which is comes under absorption spectroscopy, and that is UV visible spectroscopy. Now, here in UV visible spectroscopy, this is dependent or this is derived from electronic transition, that is the type of transition here, and from electronic transition standard absorption spectroscopy is derived for analyzing small and large molecules. So, absorption spectroscopy, if we say, it basically refers to a number of spectroscopic techniques that will analyze the absorption of the energy of the radiation by a sample. The absorption by a sample may be defined as the fraction of the radiation absorbed by the sample from the pool of incident radiation falling on it. Now, the absorption is basically determined by the atomic and molecular composition of the material. So, the absorption will occur at frequency that has the energy equivalent to the difference between the two quantum mechanical energy states. The energy absorbed causes the electrons to jump from one energy level to the other, and absorption brings about quantum mechanical changes, which could be say as we said electronic level, it could be vibrational like in IR spectroscopy, rotational could be there, but that we are not going to discuss. So, this could be different kinds of transition could occur in these molecules or atoms, and which will give you a lot of information for analysis. Let us start with the first technique, that is UV visible spectroscopy. Now, this branch of spectroscopy deals with the interaction of matter with radiation in the ultraviolet and visible region, and the transition involved in this form of spectroscopy are primarily electronic transition, as we have discussed. If you could recall, UV visible and part of little bit of infrared and X-ray comes under electronic transition. It refers to, so here it refers to absorption spectroscopy, or also it could be reflectance spectroscopy in the ultraviolet visible spectra region. This means that it uses light in the visible and adjacent near UV and infrared ranges, that is both sides of the visible range actually. The absorbance or reflectance in the visible range directly affects the perceived color of the chemical involved, and this technique is complementary to say to fluorescence spectroscopy, where fluorescence deals with transitions from the excited state to the ground state, and while absorption means transition from the ground state to the excited state. So, both are like complementary to each other, and in fluorescence there is a light emitted. So, that could be seen actually, mostly it is emitted in visible region. Now, in this particular, like as we were talking about electronic transition, molecules containing say sigma or pi or non-bonding electrons can absorb the energy in the form of ultraviolet or visible light to excite these electrons to higher anti-bonding molecular orbits. And like I said, the energy should be equivalent to the difference in the energy of two states. Types of electrons and their possible transition could be as shown in here. If you see in this figure, then you can see there could be different types of transitions. So, one is sigma bonding, which is the main bonding actually. Now, in sigma bonding, the transition is only from sigma to anti bonding orbital, that is here. There could be other transitions like from pi electrons or pi bonding to the anti-bonding or pi anti-bonding orbital. Then there could be other transitions, that is from non-bonding electrons, that is n to sigma or n to pi anti-bonding. So, these are a few transitions, those are sigma to sigma anti-bonding, then pi bonding to pi anti-bonding, then n, that is non-bonding to sigma anti-bonding, and from non-bonding to pi anti-bonding. These could be different types of transitions which could occur while absorption of the energy in the form of ultraviolet or visible region. Now, to take a few examples here, from sigma to sigma bonding to sigma anti-bonding transitions, this is for compounds with sigma bonds only, and it requires high it has a high delta energy or difference in the energy of the two states here. So, it requires a wavelength which is smaller wavelength or high frequency. So, this will be around less than 200 nanometer wavelengths are usually does, but it is not it occurs only in saturated hydrocarbons with sigma orbitals like cyclopropane, cycloalkane, to take a few examples here. So, this is like kind of not so common actually. The second transition which we were talking about was from non-bonding orbit to the pi anti-bonding transition. Now, the excitation of electrons from non-bonding orbital such as unshared pair of electrons on say nitrogen, sulfur, oxygen or any other atom to an anti-bonding pi orbital. It usually occurs in double bonds where heteroatoms like for example, CO or C double bond O, C double bond S or N double bond O are there. Now, this is mostly asymmetric and forbidden, and it is a low intensity transition. It occurs only saturated aldehydes and ketones, and lambda max is around 185 to 300 nanometer. Then there could be from bonding pi to anti-bonding pi transitions, which occur in compounds containing double, triple or aromatic rings. A pi electron is excited to an anti-bonding pi orbital, and this is like symmetrical and allowed, and it is a high intensity transition. It has if there is extended conjugation, it will lower the difference in energy and will increase the lambda max. Then there is transition of non-bonding electrons to sigma, which is sigma anti-bonding transition. Excitation for non-bonding to anti-bonding orbital is also like it could be occurring in only few like say CH3OH or others. Now, so these are a few transitions which could occur between different kinds of in atoms or molecules. If we consider macromolecules like say proteins or DNA, which are quite studied in biotechnology. In protein, dominant chromophore is the amide group, which has N that is non-bonding to anti-bonding sigma transition at about 220 nanometer, and has an intense pi to pi star, or we can say bonding pi to anti-bonding pi transition at about 195 nanometer. Electronic transition of most side chains occur below 200 nanometer, and it is overlapped by the previous pi bonding pi to anti-bonding pi transition of amide group, except for certain amino acids like phenylalanine, tyrosine or tryptophan, and also cysteine, methionine and disulfide groups, which will have their electronic transitions below 300 nanometer. And as we know that phenylalanine, tyrosine and tryptophan has pi electron systems, and they have this bonding pi to anti-bonding pi transitions. In nucleic acids, bases are the chromophores, which have extensive pi electron systems and which give rise to pi bonding to pi anti-bonding or pi star transitions. In proteins, like mostly absorption occurs, or protein absorbs at 280 nanometer, and as we have seen around 220 nanometer because of amides, and around 280 nanometer because of these aromatic residues actually, or aromatic side chains. Likewise, the lambda max for nucleic acids is around 260 nanometer, and due to the bases here. So, this was about little bit about different types of transitions which occur in molecules or atoms, and particularly as we have seen in proteins and DNA. Now, UV visible spectroscopy, as we see, is primarily governed by the Beer-Lambert's law. As we see here, molecules absorb light, and as the molecules absorb light and interact with electromagnetic radiation in this form, this absorption can give information about structure or chemical composition or concentration of the absorber, and is a very powerful technique for analysis for small and large molecules. So, here Beer-Lambert's law, which is a combination of two laws, as we know. One is Beer's law and another is Lambert's law. One deals with concentration of the absorber, another with the path length. So, both of these laws are dealing separately with the absorption of light, one related to concentration of the absorber, that is the substance responsible for absorbing the light, that is called absorber, and the path length or thickness of the layer, that is what is the path length of the absorber it is present. So, it states that the absorption of a solution is directly proportional to the concentration of the absorbing species and the path length. Thus, for a fixed path length, most of the time cuvettes are of a particular size. So, they have a fixed path length, say of 1 centimeter or so. For a fixed path length, UV visible spectroscopy can be used to determine the concentration of the absorber in a solution. So, it states that the concentration of a substance in solution is directly proportional to the absorbance of the solution. Remember the instruments which measures this like a spectrophotometer, there will be a limit to this linear or direct proportional region actually, but very high concentration this linearity will be lost. So, this has to be remembered that at a particular concentrations these measurements has to be taken. So, if you express mathematically the Beer's law, it will be like absorbance, like I said is proportional, and it is expressed in terms of here that is molar absorption coefficient or molar extinction coefficient, as we can call it, the concentration, and L is the path length here. For each species or molecule, the molar absorption coefficient or molar extinction coefficient refers to the particular it is a characteristic of that particular substance in a particular solvent, and also it at a particular wavelength actually, like it is clearly written, it is for particular wavelength. So, if you see here this is how this is a cuvette where you have a particular absorber in a solution. It has a the absorber has a particular of a set concentration, and there is a path length here. As the intense light, that is initial light, enters, it is absorbed to a different extent, to varying extent depending on the concentration and type of molecule in here or absorber here, and there will be. So, this is incident light and this is the transmitted light which comes out after certain absorption which occurs in here. So, when sample molecules are exposed to light having an energy that matches a possible electronic transition within the molecule, some of the light energy will be absorbed, and as the electron is promoted to higher energy orbital, and this will be recorded by a spectrophotometer, and it will record the wavelength at which this absorption occurs, together with the degree of absorption, that is height of the peak you can say at each wavelength molecule, that show increasing degree of conjugation requires less energy for excitation, and as a result absorb radiation of higher wavelength. So, depending on what kind of molecule is in there, it could be at higher wavelength or lower wavelength, but it is typical of or characteristic of a particular kind of molecule. Now, as we were discussing, it is a particular wavelength where absorption is maximum is called lambda max, and if we consider in simple terms, there should be spectral lines which should be obtained at that particular transition where it is occurring, but most of the time it is quite complex, and there are a set of spectral lines. As I have shown you in the energy diagram, there are vibrational labels and others. So, to understand simply, it is like set of spectral lines which are there, and so you do not really get to see the spectral lines when the output is shown. Rather, what you see is a peak actually, a small peak depending on the concentration, it will have a particular area, and that peak is shown in form of absorbance on the readout. There are a lot of things to be considered for Beer-Lambert's law. The bandwidth utilized for the analysis should be sufficiently low or it might alter the absorption features of the sample. The absorption should be taken at the wavelength where the absorption should be the highest, at the variation might lead to alteration in the measurements. The light that is used should be of a monochromatic wavelength. Then concentration of samples should not be too high, as it leads to saturation of the absorption signal and might result in false, might get false results here. So, remember the molar absorption coefficient will alter, or the Beer's law will not will be violated when you have a very large concentration, or different aspects needs to be met, so that Beer's law is not violated. Like here, absorption should be taken at particular wavelength. Many times a molecule can have multiple lambda maxes for different properties, chemical properties of the absorber. So, those things also needs to be taken into account actually. So, when we are considering these things in terms of lambda or lambda max, or in terms of like I said, the spectral lines are obtained, but like let me show you that on a screen here. So, particular transition should be obtained in terms of, if I say in terms of spectral lines actually, but like I said, there are it is much more complex, because there are a lot of vibrational labels and how it goes from, like it is not exact, it is like it will absorb at, and transition takes place, which is much complex. So, you do not get spectral lines as such, but you get series of spectral lines which are closely spaced in here. So, what you get in real terms, you get a peak actually, which does not show, or to be very clear, you get a peak here, which is which does not show these spectral lines, but there are closely related or closely spaced spectral lines in here. The absorbance, if I say, or optical density as we say, as the readout comes out, it is or a spectrophotometer is in essence reading the absorbance at a particular wavelength. So, if you plot absorbance versus wavelength, then you will get these peaks at particular wavelength, and that is lambda max in here. So, that has to be understood, that as we were talking about Beer's law, one is concentration, if it is too high, then also it will be violated. Then at proper wavelength it has to be done, so that Beer's law hold, and lot of other precautions has to be taken, so that this particular analysis could be done without any problems, or without any spurious results actually. Let us discuss little bit about the instrumentation, as we have discussed about principle of the UV spectroscopy, that is based on Beer's law or Beer's Lambert's law, and how this is an electronic transition here. Let us discuss about the instruments which are utilized for measuring this absorption. Now, if a particular instrument is only based on like wavelengths in visible region, that is colored region, then colorimeters could be used, but more widely used and sophisticated instrument is spectrophotometer. So, the UV visible spectrophotometers are widely used, and they measure the intensity of light passing through a sample, and they will compare it to the intensity of light before it passes through the sample. So, as I have shown you earlier, this like as light enters, it is an incident light, and when it passes through, it is a transmitted light. So, the ratio here is called the transmittance, and the transmittance will be expressed as a percentage. That is, if there is no absorption, it is the 100% transmittance, and if there is complete absorption, there is 0 transmittance, that is an object is totally opaque. There could be UV visible spectrophotometer and could be configured to measure reflectance also. In this case, the spectrophotometer measures the intensity of light reflected from a sample, and it compares to the intensity of light reflected from a say reference material, and the ratio will be called the reflectance, and usually expressed as a percentage reflectance. So, this is how either transmittance or reflectance could be measured. Now, the basic parts of a spectrophotometer are essentially a light source, then a holder for the sample, that is cuvette or where you can put in your sample, then a diffraction grating in a monochromator or a prism to separate the different wavelengths of light, and finally a detector. So, let us discuss each of these. A light source could be, or the radiation source is often a tungsten filament which is from 300 to 2500 nanometer, mostly towards visible region actually, and its cutoff is around 350 you can say. Then for UV visible, so visible if you take tungsten filament, then for UV a deuterium or arc lamp is required, which is a continuous over the ultraviolet region from 190 to 400 nanometer. Both lamps could be are switched on when it is working, or either could be switched on. Then xenon arc lamp, which has a continuous range from 160 to 2000 nanometer, could be utilized in place of above two lamps, that is tungsten and deuterium. Or more recently there are light emitting diodes for the visible wavelengths, which are also utilized. Since the emitted light consists of many different wavelengths here, a monochromator consisting of either a prism or a rotating metal grid of high precision called grating is placed between the light source and the sample. So, that is also required to separate different wavelengths of light. The detector is typically a photomultiplier tube, or it could be a photodiode, a photodiode array or charged coupled device. Single photodiode detectors and photomultiplier tubes are used with scanning monochromators, which filter the light, so that only light of a single wavelength reaches the detector at one time. The scanning monochromator moves the diffraction gratings to step through each wavelength, so that its intensity may be measured as a function of wavelength. Fixed monochromators are used with CCD or charged coupled devices and photodiode arrays. As both of these devices consists of many detectors grouped into one or two dimensional arrays, they are able to collect light of different wavelengths on different pixels or groups of pixels simultaneously. So, there could be various arrangements here in these sophisticated instrument. Now, spectrophotometer can be there could be specialized spectrophotometers which have various options. Like one is spectrophotometers can be either single beam or a double beam spectrophotometer. So, the single beam spectrophotometer focuses all light on the sample, and you have to do reference separately. In a double beam spectrophotometer, light is split into two before it reaches the sample, and one of the light paths is directed to the reference, where the other goes through the sample. So, there like it is divided into two, one goes to the sample, other goes through the reference cell to be analyzed, and the intensity ratio between the two is measured. So, mostly what is done when you have a single beam spectrophotometer, baseline correction is done with the reference, that is zero absorbance is taken at the reference, and then or you can say it is zeroed, and then the sample is absorbance is taken. And double beam, the reference automatically sets the baseline. In a double beam spectrometer, therefore, the light is split into two beams before it reaches the sample, and like one is going to reference, other going to the sample. The reference beam intensity is taken as 100% transmittance, or we say zero absorbance, that is it is zeroed actually. So, that the peak starts from the very baseline in terms of if it is passing through the sample. So, the measurement is displayed as the ratio of the two beam intensities. Some double beam instruments have two detectors, photodiodes, and the sample and reference beams are measured at the same time. In terms of sample holder, Samples are typically placed in a transparent cell, and these transparent cells are known as cuvettes. Cuvettes are typically rectangular in shape, and they come in different sizes and different path lengths. Most of the time, very commonly used one has an internal width of 1 centimeter. The most widely applicable cuvettes are made of high quality silica or quartz glass, particularly for UV region, because these are very transparent throughout the UV and visible region. But for visible region, you can use glass and plastic cuvettes could also be utilized, are very common. Although glass and most plastic will absorb in the UV region, so they are not utilized there. So, for UV region you require say quartz or silica cuvettes, whereas for visible region either could be utilized, that is plastic or quartz cuvette. This is a schematic of spectrophotometer. These are two here, if you can go through. There is a light source, as we have discussed, this could be different light source. There is a filter or monochromator. Filter is mostly utilized for again filtering the wavelengths, and particularly in the it is utilized in colorimeter for different color filters are available, but in spectrophotometers mostly monochromators are used. Then there is a reference cell, there is a sample cell, and like I said, there could be two kinds. One is single beam or double beam spectrophotometers. There is a photo detector, amplifier, and then finally readout in the form of peaks will be shown, that is absorbance. More is the absorbance, bigger will be the peak at a particular wavelength. Then here it is a double beam spectrophotometer. You can see it is the same, but light is here divided into two through a beam splitter, and one beam goes to the reference cell, another goes to the sample cell, and then finally difference amplifier is there and readout is taken, like it is for reference cell it is 100% transmittance and 0 absorbance is taken. So, this is a typical single beam and double beam spectrophotometers. There could be different like alterations in modern spectrophotometers for various applications. There could be attachments for say very small cuvettes, like replaceable cuvette holders are there. There could be a provision for taking absorbance at very low temperature, so that thermal motions are avoided. There could be provisions for stopped flow kinetics, and there should be a lot of different types of analysis could be done in these spectrophotometers. So, there are a lot of advancement which has taken in here. Now, if we consider as far as in visible region relationship between light absorption and color. So, white light when falls upon a sample, the light may be totally reflected, in which case the substance appears white. If the light is totally absorbed, I think it will be, as we all know, in which case the substance will appear black. If however, only a portion of the light is absorbed and the balance is reflected, the color of the sample is determined by the reflected light or reflected wavelength. Thus, if violet is absorbed, the sample appears yellow green, and if yellow is observed, sample appears blue. Likewise, so the colors are described as complementary here. However, many substances which appears colorless do have absorption spectra, and in this instance absorption will take place in the infrared or ultraviolet region, as we have discussed. Now, this is a table here which you can see that there are different color absorbed. The color, if this particular color observed, you will see a particular color observed here, and which are absorbance wavelength is given in here for various different colors or different wavelengths in visible region actually. So, this only depicts about if the particular say for violet is absorbed, then yellow green will be seen, and this is the range which is absorbed actually. Likewise, the whole range different situations are given in here. So, this was about the spectrophotometer, which is now modern instrument with various modifications and advancement, and for so that lot of different arrangements or different accessories could be put in, and lot of applications could be run on these spectrophotometers. There are a lot of applications of UV visible spectroscopy, and we will be discussing a few here. Like we said, it could be utilized for analysis of small and big molecules and for various applications. So, UV visible spectroscopy is routinely used in analytical chemistry for the quantitative determination of different analytes. For example, say in transition metal ions, highly conjugated organic compounds, biological macromolecules could be routinely checked in for. Most commonly this type of spectroscopy is used for quantification of biological samples, either directly or by colorimetric assays. There are a whole lot of different kinds of colorimetric assays which are available for protein and DNA. In many cases, proteins can be quantified directly using their intrinsic chromophores like tyrosine and tryptophan, as we have discussed. Tyrosine and tryptophan absorbs and protein therefore absorbs at 280 nanometer, and DNA or nucleic acids absorbs at 260 nanometers. Spectroscopic analysis is commonly carried out in solutions, but solids and gases may also be studied. Solutions of transition metal ions can be colored, they absorb visible light because of d electrons within the metal atoms, and can be excited from one electronic state to another, and the color of the metal ion solution is strongly affected by the presence of other species, say certain anions or ligands. For example, the color of dilute solution of copper sulphate is very light blue, but if you add ammonia, it will intensify the color and change the wavelength maxima or lambda max, which is the maximum absorption. Organic compounds, especially those with a high degree of conjugation, for example, DNA, RNA or protein, will absorb light in the UV or visible region of the electromagnetic spectrum. The solvents for these determinations are often water, for water soluble compounds, or it could be certain organic organic solvents. Organic solvents may have significant UV absorption, and so not all solvents are suitable for UV spectroscopy. Like for example, ethanol absorbs very weakly, so that could be one. Solvent polarity and pH also affect the absorption spectrum of an a particular molecule. For example, tyrosine for example increases in absorption maxima and molar extinction coefficient when pH increases from 6 to 13, or when solvent polarity decreases. Let me show you that on your screen. So, these things like I said, pH or polarity or orientation play a major role in here. For example, if I say pH, in terms of pH actually, if we are having say molar absorption coefficient in here or epsilon, which is in terms of particular units, and if say just to show you here, and if there is particular wavelengths here, now if I consider that the two pH, which is pH 6 and pH pH 6 and pH 13 as I was talking about, then there is a particular difference actually here. You can see that this is a tyrosine spectra, absorption spectra of tyrosine. This is as pH 6 and this is at pH 13. Here the absorption maxima is around 274 nanometer, and here the lambda max is around 295 nanometer, somewhere around that. So, what you see is, and also there is an increase in the absorption coefficient. So, when it is done at two different pH, the pH affects the lambda max as well as molar absorption coefficient or extinction coefficient. Likewise, it also affects what you call polarity, or the what you call orientations will also affect the thing. For example, if say one needs to do solvent polarity here, if solvent polarity has to be looked into, let us see how it affects the absorption. So, in one case it will be, supposing this is the these are not very exact figures, but you can at least rely on them for understanding. So, these are two spectra in here. One is at ethylene glycol, which is non-polar, and another is in water actually. So, you have two of these spectra here, you can see. Now, one spectra is in polar and another is in non-polar solvent. You can see there is a lambda max increase in here. The lambda max is increased in case of the ethylene, in case of less polar solvent. So, polarity will affect certainly the lambda max and also molar absorption coefficient. And third part we were talking about is the orientation effects. So, orientation effects, that is how a molecule is present in a situation. If we take optical density in here and say and this example we are taking is of DNA, and I think all of you know that DNA is present in different forms. So, there are three curves I have made. Now, if you take this one, this is double stranded DNA, that is arranged quite well and this is single stranded DNA and this is free nucleotides. So, you can see in here that as these are the arrangement changes, like in double stranded DNA they are well arranged and you will have a particular spectra, and as this arrangement is loosened, like in single stranded DNA or free nucleotides, there is increase in absorbance. So, these are a few examples where you can see that effect of pH, polarity or orientation on different spectra. So, as we have seen, solvent polarity, pH and orientation of a particular molecule in different forms can affect the absorption spectrum of these compounds. UV visible spectrophotometers, as they could be utilized for recording the absorption patterns of different molecules. They could be attached to other instruments like HPLC, where the presence of an analyte could be found out, or a response assumed to be proportional to the concentration could be recorded. For accurate results, the instrument's response to the analyte in the unknown should be compared with the response to a standard, and this is very similar to the use of calibration curves actually. So, this spectrophotometer could be arranged with a purification instrument like HPLC or FPLC for analysis of different types of purified analytes. The wavelength of absorption peaks can be correlated with the type of bonds in a given molecule and are valuable in determining the functional group within the molecule. The nature of the solvent, the pH of the solution, temperature, high electrolyte concentrations and the presence of interfering substances can influence the absorption spectrum. So, the experimental variations such as the slit width, that is effective bandwidth of the spectrophotometer will also alter the spectrum. To apply UV vis spectroscopy to analysis, these variables must be controlled or accounted for in order to identify the substances present. So, lot of procedural things has to be performed to get accurate results actually. So, finally, UV vis spectroscopy could be utilized for a lot of different types of analysis. There are a lot of different applications. To summarize, it could be utilized to determine concentration of an analyte by seeing the absorption patterns and comparing it to the standards, known standards, and through calibration curve the concentration of unknown sample could be calculated. It could be utilized for assaying a chemical reaction to observe the chemical reaction or assay enzymatic assay or other assays which are going on in a chemical reaction. As one substrate changes to a particular product which could be colored or absorbs at a particular wavelength, that could be process could be monitored. There could be structural studies of DNA could be performed, like for example, denaturation and renaturation studies could be performed, where melting curves could be drawn as a function of temperature, and depending on that one could derive the idea of about GC content of DNA, and depending on GC content there will be a particular melting temperature that could be derived from these studies. So, a lot of structural studies could also be performed, like as we have shown, depending on the orientation or depending on the particular form of the macromolecule, they will have different absorption. This UV vis spectroscopy can also be used for studies of proteins and structural studies, certain structural. Now, here when we say structure, does not mean 3-dimensional structure, but certain signature, certain strong indicators are there to understand, like for example, at different pH titrations or by difference spectroscopy, there could be certain indications about structural changes could be observed for both DNA and proteins. So, there could be a lot of different applications of UV vis spectroscopy, and lot of different kinds of analysis could be performed on this as per requirement. To summarize, UV vis spectroscopy is a kind of absorption spectroscopy. It is utilizes the ultraviolet and visible region of the electromagnetic spectrum, and as per the absorbance of the molecules in the solution, lot of different kinds of analysis could be done. It follows like here it is governed by the Beer-Lambert's law, and the instrument which is utilized is it is spectrophotometer, which have been modified and advanced quite a lot for different kinds of analysis, and a lot of different accessories have been included to do experiments, say at different temperature or in different conditions. So, this is one very useful analytical techniques in the field in the area of biotechnology, and it is utilized for analysis of different compounds including proteins and DNA. In the next lecture, we are going to discuss a few more techniques which are based on absorption or emission or other different phenomena. Thank you for your attention.